In individuals who have DMD, dystrophin is no longer present, which decreases the memory stability in skeletal muscles. This significantly impacts the muscle and motor function of these individuals, decreasing their quality of life. Individuals eventually end up wheelchair-bound and end up dying prematurely due to cardiac and respiratory failure caused by DMD. This is how DMD affects the motor skills of individuals. But how does DMD affect patients in terms of language and neurodevelopment? Well, a study in 2015 looked at these neurodevelopmental rates using a sample population of 130 boys with varying mutations of DMD. The study was conducted in Europe and consisted of boys from the UK, Italy, and Germany. The study used 5 measures to determine if there were any disturbances present. The first being the Social Communication Disorder Checklist, or the SCDC, the second being the Wechsler Intelligence Test for Children, and the third being the Child Behavior Checklist, the fourth being Developmental Diagnostic and Dementia Interviews, and the fifth being the Connors Parenting Rating Scales. Based on the information obtained from these tests and the significantly elevated SCDC scores, we saw high rates of neurodevelopmental disturbance. 26% of the boys were perceived to have some sort of intellectual disability, 21% of the boys scored above threshold diagnosis for the autism spectrum disorder, 44% tested for inattention, and 24% had hyperactivity.
Another study published in the Journal of Pediatrics compared a sample size of 130 patients with DMD to their unaffected siblings, which numbered 59. They used siblings to control for genetic, familial, and socioeconomic variables. The participants of the study were selected from Atlanta and New York and were between 4 and 14 years old. The study monitored 10 different milestones, including smiling, sitting, crawling, standing, walking, saying single words, constructing complete sentences, reading, and becoming bowel and bladder trained. The results of this study showed that a greater percentage of DMD patients than their normal siblings rated low on language milestones, such as speaking their first word and speaking in full sentences.
These results, however, only apply to some of the DMD patients, not all. Because the researchers did not control for the varying mutations of DMD, and these delays in language development aren't seen in all patients with DMD, it begs the question, is there a specific mutation in the dystrophin gene that leads to these delays?
Dystrophin protein and several dystrophin isoforms are expressed in skeletal muscle, cardiac tissue, and the brain. Boys with mutations downstream of DMD exon 45, which specifically affects the brain-specific dystrophin isoform DP140, have structural brain abnormalities and deficits in performance-based psychometric measures. Dystrophin protein DP140, which uses DMD exon 45 as a unique first promoter, is highly expressed in the human cerebral cortex and cerebellum, regions that are critical for language and executive function. Also, dystrophin 140 was recently shown to be expressed by oligodendrocytes, and abnormal myelin development was seen in the MDX mice models of DMD. Speech delay is more common in boys with mutations downstream of exon 45. The average age of speaking in boys with mutations upstream of exon 45 was 26.7 months, while boys with mutations downstream of exon 45 was 30.3 months. While lateralization of language networks in the brain occurs in infancy, the pathophysiology of speech problems in DMD remain unclear, and specifically how the absence of dystrophin affects speech is largely unclear. Full-length dystrophin and shorter dystrophin isoforms may have distinct roles during brain development, such as synaptogenesis and axon guidance. A better understanding of the neurobiological mechanisms by which dystrophin and its isoforms influence brain development and function could lead to personalized interventions and improve neurodevelopmental outcomes in DMD patients. There is conflicting research regarding DGC's true impact on brain tissue. However, investigators have reported brain abnormalities ranging from negligible to severe. These abnormalities include neuronal loss, neurofibrillary tangles, Purkinje cell loss, dendritic abnormalities, disordered structure, and even gliosis. MDX mice, researchers have reported substantial differences between the gross brain structures of MDX mice compared to the control. The absolute number of cells was diminished by 50%. Cell packing density of cortical LIR5 neurons was higher in the MDX than in the controls. While the cell packing density of the corticospinal neurons was lower in the MDX mice than the controls, corticospinal cells constituted 50% of the neurons present in layer 5 in the controls, but only 35% in the MDX mice. In MDX mice, the hippocampus and cerebellum, there was a substantial reduction in GABA A clusters. The number of GABA A clusters was reduced by approximately 50% in MDX mice. Deficits in verbal working memory, a large component of the DMD cognitive deficit, are now thought to be having a cerebellar focus.
Research on this topic is still fairly new, but we hope to learn more in the coming years.